Welcome back, ladies and gentlemen. I hope the break provided you with some time to connect and relax. Up next, we are excited to introduce our second speaker of the day, Professor Yann Boucher, who will be delivering his presentation, One Ocean Health, Connecting Climate, Environment, and Health. Professor Boucher is professor at the NUS Salk Neval School of Public Health and principal fellow at the Singapore Center for Environmental Life Sciences Engineering, CELSI, at NTU. He is an expert on the ecology and evolution of enteric bacterial pathogens, both inside and outside the cells. In today's talk, Prof. Boucher will share about how some of these bacteria live and interact with marine environments and organisms, and the implications this has for our health and the oceans. Without any further ado, Dr. Boucher, please.
Thank you very much for the intro. Is that the pointer? Let's get to it. Yeah, thanks very much. Thanks so much, Yanni, for inviting me. I'm a bit of the odd duck out here because I work on public health, but I'm a microbiologist by training, and I've always dealt a little bit with aquatic bacteria and waterborne diseases. This is I don't know if any of you have seen this in some of the bus stops in Singapore. Anybody seen that? All right, so this was an advertisement campaign by the government. I think they were testing dramatic scenes and the effect on public receptivity about climate change and rising ocean levels. Um, and I remember when my daughter saw that on the bus stop, she started crying and she's like, "Are we gonna die, Papa?" And I was like, "No, we're okay. We live on the ninth floor. We'll be fine." Um, but it does illustrate that the government's very aware of some of the issues with climate change and rising sea levels is one of them. Why is it? Down. Oh, I see. Go down. Okay, great. So one thing I just want to advertise first is that actually I'm the co-lead for this new program in public health. It's called Climate, Environment, and Health. Um, and the lead is Kimberly Fornes, and she actually has lived in Borneo for a very long time before moving here. And she actually, I realized that she works in Sarawak with the same people that we're presenting. That she worked with a center for it's a new center for infectious diseases down there. And actually, one of the main projects that we're gonna start doing in one of our flagship project is actually to um, to pick different spots in Southeast Asia as hotspots for climate change or environmental change and the impact on human health. And one of the spots she's discussing with her colleagues is in Kota, actually in Sarawak. So what we want to do with these projects is actually do a close monitoring of different health parameter for people that live on the coast, both disease they're carrying, wearing monitors, wearable for monitoring different physiological characteristic regarding heat and things like that, but also couple that to environmental monitoring like landscape changes, pollutions, algal bloom in the ocean. So she takes care of the terrestrial side, and I take care of the water side of things in terms of this monitoring. So this is a work in progress, and I'm quite keen to talk with some people in the audience about setting up some of these from these hotspot monitoring around Southeast Asia. Um, so today, I want to talk about a lot of what I'm going to talk about is about how climate change is affecting infectious diseases, right? And waterborne diseases and those that come from the ocean or in aquatic environments. And one of the things that is really unique, because I used to be in Canada before and I always worked in Bangladesh a bit, but I always found, that there's quite a lack of data in equatorial and tropical areas in terms of infectious diseases, especially waterborne ones. In North America, there's a lot of data in Europe collected on those diseases, but not so much near the equator where we are here. And it's actually really, really interesting because there's a completely different ecology for microorganisms that live in the water. Now, one of the things I study is vibriosis. Okay, and vibriosis is infections you get from these bacteria that live in the ocean. Are called vibrios, and they can have a lot of different symptoms. They can come from like diarrhea to septicemia to even necrotizing fasciitis, where you have to flesh-eating disease. So I'm not going to show any pictures because it's gross. But it can be quite dramatic, right? And people have been studying this for a very long time, but there's actually not that much that has been done in the area here. And so there's these different pathogens. Okay, so you don't really need to lick the list, but you might be familiar with some of them. Vibrio cholera was one of them. I'm going to talk about that later about the disease cholera, but vibriosis is mostly not cholera. an infection you get when you're eating seafood or you're swimming in the water and you have a cut, an abrasion on your skin. Okay, and all of these different species can cause these different types of infection. And some have more tendency to be in food, some more in cuts and wounds when you're in the water or you swallow water. So it varies, but they can all cause a range of symptoms. Um, and I've been recently to a workshop. Actually, I found there's a lot of things in common with other people. I've been to a workshop in the Quadram Institute in the UK, where I met with Craig, who's in CFAS. Okay, we have some people from CFAS here. And these are all experts. Also work with the CDC in the USA. And the USA actually happens to have the best reporting system for vibriosis in the world. And they've worked for a very long time to develop this, and it's a mandatory reporting of all the infections. Any states in the US that get these infections, it gets sent to the CDC. The genome of the bacteria gets sequenced. It goes in the database, and there's a lot of detail. There's like so much detail. One of the cases is somebody that was doing windsurfing that got struck by lightning. They fell in the water. The ends were burned, and then they got a Vibrio infection in their hands. So they go into that much detail. Now, when I moved to Singapore, I wanted to see, do we have any databases like that? Because Vibrios are actually extremely correlated with climate change. So basically, their abundance in the water is directly correlated with water temperature. So which makes them a prime target to look at the impact of climate change on disease, right? Because there's more abundance, more times you reach the infectious doses, more infections. And now I'll show you some statistics. Infections are rising in temperate areas. Um, and this is a bacteria that grows extremely fast. It has some of the fastest growth in all of bacteria. Some of them have 10-minute duplication time, which means they can grow so fast. It's hard to imagine for us. And this means they can respond to nutrient changes, temperature change very rapidly, and bloom, and then be a hazard for people that come in contact with the water, right? Um, so recently, people are starting to analyzing what factors contribute to vibriosis in terms of the risk and calculating the risk. So these bacteria, you have to look at their ecological suitability to see if there's a possibility of infection, right? The salt level matters. Some species love the salt, like most of the vibrios, they love salty water. Some of them, they like it more brackish or freshwater, like Vibrio cholerae. But now with climate change, the zone in which these bacteria can grow has been expanding. Okay, especially in temperate climate like Europe, for example, and North America, this has been expanding. So this is something we must monitor. And all these marine stations we're talking about could play, a crucial role in terms of getting more monitoring. And there's also all sorts of phenomena that spread these bacteria around the world because these are waterborne bacteria. So whenever there's ballast water from a ship that they take water in one port and go and dump it somewhere else. In Singapore, we have all these, you just have to have a look, right? I don't know if the visitors have been to East Coast or West Coast Park. You just stand there and you see hundreds of ships in on the coastline, right? They're all dumping their ballast water for somewhere else in the world. So we get a mixture of microbes from all around the world. So we can import disease that way. Aquaculture is another one, right? When there's a trade, you're growing some oysters or some mussels or something, and they're being moved to different countries because they're productive type. They can carry their pathogens with them. And there's even some hypothesis that marine currents could actually carry some strains of pathogen across the ocean from one continent to another. So, for example, there's some hypothesis that some pathogenic Vibrio cholera were carried from Asia to America through the equatorial current, right, during an El Nino event. So these are all possibilities that we need to think about when we think about, how these strains go around the world and how they can cause outbreak in multiple different places. Now, once they get to a new place, they have to adapt, right? Because it's not the same water temperature. Some places have seasons, some places like here, not so much, right? So they have to compete with the local bacteria. So it's not necessarily that easy to establish themselves, but it can happen. And then we have to look at all the exposures and risks that vary from country to country. For example, the age of the population, how many people live on the coast? Do they eat a lot of seafood? What type of seafood do they eat? Does that make them more likely to get sick? So all these factors come into the calculation of evaluating the risk. Um, and as I was mentioning, these are just projection, right? So um, the different climate change scenarios are illustrated here. Okay, some more drastic than other, and this is calculating the amount of coastline that is going to be appropriate for Vibrio infection, how it's going to increase with time, right? So right now, coastlines are, in warmer areas, they're more likely to have a Vibrio infection, but this is going to change. The amount of coastline is going to increase. You can look over here, right? In Europe, some drastic increase. Some bays in China also might have a drastic increase in terms of being. There's a possibility of getting vibriosis. So this is a changing dynamics that we're actually observing from year to year. Um, and in terms of we were talking around the world that different marine station collecting data, well, studies on the effect of climate on waterborne infection also are unevenly distributed around the world. So this is just an overview that I've done with Craig, of how around the world, which countries, and I've done studies on incidence of vibriosis, and the impact of climate on it. And if you can look, oh, there's a lot in North America, there's a lot in Europe, but Southeast Asia, one study for the whole of Southeast Asia. I mean, this is crazy. We've got hundreds of millions of people, right? And what is special also is that Southeast Asia is completely different ecologically from North America or Europe. So we need our own studies here, right? So for example, it's predicted that the amount of Vibrio bacteria, the amount of coastline is increasing, like I showed you, in warmer areas, right? But in the tropics, it's predicted to stay flat because the water is already warm here, right? So you have to use different models. So when just another example also of the effect of climate. So this is something it's in 1997. There was an El Nino event, okay, and there was we've heard a bit about marine heat waves. So this is an example of one, right? There was an event of the west coast of North America where there was a mass of warm water, which is usually more towards the south, right? That moved north all the way to British Columbia and Canada, also Oregon and Washington State. Okay, and this is the number of cases in Washington states of vibriosis that happen. And you can see the timing in July and August when that water mass is moving north, the cases that are happening north here are rising. And then when the water mass goes away, the cases decreases. So this is like the tightest connection with climate that you could ever imagine, right, for a disease. So some diseases are intrinsically linked with climate and the ecology that's happening in the water. Another example here, it's Hurricane Katrina in 2005, and as we speak, there's Hurricane Milton that's also going there right now. So this might happen again. So this is when they monitor the number of cases of vibriosis, right, in the Gulf Coast. So this is this massive base in south of the United States between Texas and Florida, and you can see the hurricanes going through with the different index of how strong it is in different places, and each county is colored by the number of cases of vibriosis they're having. And you can see that when the hurricane is going through Katrina, boom, it spikes massively, right? The number of cases. So maybe this is going to happen again with Milton. And as these storms becoming more frequent, you'll got more of these bumps in diseases, and we need to be prepared for that. This is another example. It's quite amazing. And from year to year, they're measuring what's the northernmost cases of vibriosis that you're getting in North America, right? And as the year progresses, you can see that this line is moving north and north and north and north, right? So 2018, and I know that in the last few years, there's been cases in New York State, Massachusetts. It's moving even more north, right? So as the water warms, this is the increase in coastline that's susceptible to these type of vibriosis. So there's more of a threat to human health in a longer area. And there's massive population centers over here, right? So that means that a lot more people are going to be exposed to this. So when I first came to Singapore, I'm like, wait a minute. Vibriosis is related to water temperature. Why isn't everybody here dying of vibriosis? This doesn't make sense. The water is between 28 and 32 degrees. And it's like, I want to know more about this. But everybody I spoke to was like, not important. No, no, like I've never heard of this disease. So I actually went to the hospital, and my people went to the hospital, knocked to every single door because this is not a reportable disease in Singapore. Only cholera is a reportable disease. And cholera, most of the cases are just imported from people moving in. So I went and knocked on hospitals' doors. We knocked every hospital's doors. About 7 out of the 10 hospitals said yes to give us some data. So we collated the data, and what we found was quite interesting. So this is data from the U.S. Okay, and you can see that when you break it down. Oh, sorry. When you break it down by species, not all the species are changing the same in the US. You can see they're all increasing. But if you look at species like Vibrio cholerae, and this is not the one that causes cholera, it's the other ones that cause opportunistic infection, mostly gastro and wound infections. And Vibrio fluvialis are the two fastest rising out of all of them. So ecologically, they're not responding the same to the weather changes. And in and you can see that if you calculate from year to year how much the number of infections is changing, and you average it for between 2015 and 2019, you can calculate this ratio. And 1.13 means that every year there's about 13% more cases in the last 5 years. Not the last 5 years. We stopped at 2020 because COVID totally messed up their numbers in the US because they stopped collecting data. But you can see that Vibrio cholerae, for example, the 20% increase a year. This is an insane amount of increase in vibriosis. Um, and there's also a seasonality to it, right? So the species do not have the same amount of seasonality. So this is because they're related to the temperature of water. And in North America, water obviously is warmer in the summer. So you can see that the peak in infection occur mostly July, August when the water is warm, but some of them, it's a broader distribution, right? It includes late spring, early fall, like Vibrio alginolicus, but Vibrio paramolyticus is extremely tight. It's mostly all the cases in July, August, right? So all of these species have a different ecology. They don't have the same hot season, can be tighter or broader, so different temperature tolerance. And not all of them are increasing at the same pace with climate change, which means that some are more responsive than others to temperature, right? And the CDC has recently come out with this amazing dashboard where they report all the cases, and you can just go to this and you can click, and it's interactive, and you can select the species you want to know, and you can look at how many cases there are. Um, so you can get all the information. And I think this type of tools that we're talking about for marine data should also be used for waterborne infection. They're very, very useful, and they can give a lot of guidance. And so I'm working on possibly developing one of those for Singapore, and the CDC is very happy to share their platform to do this. And I think if we can do it even all across all Southeast Asia, it would be even better. So these type of data aggregation, I think, is very important, and it was mentioned before for environmental data, but I think we should couple it to health data as well. Um, so what about Singapore? I was mentioning the water is warm here, so this is just how it varies between 28 and 32, 3 different years. There's a bit of effect of the monsoon that with the temperature changes. So what happens here, right? There's been very few studies. So one thing that we found out is that when we look at the incidence, from the hospital data, is that in the US, as I showed you, it's rising for all these different species. In Singapore, it's actually pretty flat. This makes sense because um, the water temperature is not increasing as fast here as it is in temperate areas. So the infections are flat. But what is interesting actually is that we actually have about 3 times as much infection in terms of incidence that the US has. And here it's not a reportable disease, and it's so people are not aware that actually we have a lot more than some countries that monitor it very tightly. And another very interesting fact is that it's not the same species causing most of the infection. So the top one is also Paramolyticus. So this is a very common cause of seafood-borne gastroenteritis. But the second one we have here is Vibrio fluvialis. And in the US, they don't even consider it to be an important factor. But here, it's the second most common cause of infection. And we also have Vibrio farnesii, which in the US, it's a non-thing. There's like a handful of cases a year for 300 million people. And here we have quite a substantial amount. So you get different species causing the infection. And I want to highlight that this is very important. This was completely unknown. I was talking with SFA after I found that out, and it was like, oh, this is very interesting. We might change the way we test the seafood. We might have some assays for fluvialis, right? And this is important because not every country is going to be the same pattern of disease, right? When it's very different ecology in the equator and temperate, we can't just use the data from the US and what they considered the most important. We have to have our own data because it's not going to be the same because of a very different climate that we have here. And a very interesting thing is that when I was pouring into more details into on the U.S. data, um, I noticed that Hawaii, right, of all states, was very different from other states. And this also makes sense, right? Hawaii is the most like Singapore, right? It's an island. The water's warmer. It's a bit cooler than here, more around 25 to 28 degrees, but it doesn't vary too much year round. And their incidence is also quite high, similar to the incidence in Singapore. So maybe this can teach us something about ecology of these bacteria, how these infection happens, right? When you get a certain setup, maybe most of your population is coastal. There's more exposure to seawater, or the water is warmer. There's not a seasonality is warmer year round. So maybe this is what contributes to the higher incidence. So I think this comparative analysis of different places that share some similarities and places that are different can teach us a lot about dynamics of diseases. And I think I want to highlight it again. That's why we need data from around the world, because otherwise we're not going to understand what's causing these differences in disease dynamics in different places. Um, so um, some people were talking about gamification yesterday, so I do some gamification too. This is my game, Lockdown, right? It's a game on COVID, but we're working on another one, and my friend Anna, she's the artist. She drew this. I asked her to draw me a Vibrio color, and this is what she. I said, make it cute, like Pikachu. We need a Pikachu for our game. And I like, okay, I'll do it. And it's not quite as cute as Pikachu, right? It is an infectious bacteria. But we're working on another game that has to do with more with ecology of bacteria. So this is our first one. This is a cholera. So I wanted to mention cholera because cholera, it's people don't think of it as a marine bacteria, okay? But actually, oh, this thing came up at the same time as the other one. I wanted to show a map of the world where the population in all the blue countries was the same as the population in the Ganges Delta, just to show how dense the population was. And cholera is a disease. Actually, the bacteria is in the ocean, right? You don't contract cholera in the ocean because nobody drinks seawater, right? Unless you're diving, but that's another story, right? But normally people don't drink seawater, right? They drink fresh water. But in some places in the world, like the Ganges Delta, for thousands of years, people have been forced to drink slightly brackish water because there's 100 kilometers of coastline. You go inland, and it's all at sea level. So if you dig for a well, you often get salt intrusion, right? So in that large area, which is extremely fertile, so people were living there farming, but they were drinking brackish water. And this made me think that actually this is what drove the evolution of cholera. People were drinking brackish water. This is a bacteria that is not a freshwater bacteria originally. It comes from the ocean. Um, and they were drinking and drinking, so that selected for some that are adapted to living inside the human gut. And then it evolved and specialized to become a human bacteria that kind of spends some time in nature. But the reason it evolved is because humans came into contact with brackish water, which it was never meant to be, right? But it's just the ecological circumstances, right? So a relationship with the ocean and the salt water actually gave rise to this disease and made these bacteria evolve to live in the human gut. And for hundreds of years, this has killed hundreds of thousands of people, and it's one of major pandemic disease that is still ongoing today, right? Um, just to give you an idea of the importance of this bacteria, we don't usually think of microbes and how they affect human population on a large scale, but I can tell you they have a massive effect. So if you go to the Ganges Delta and you look at blood group, right? Um, Cholera has a preference in infecting people with a specific blood group. So you see here is the breakdown into the percentage of people in each of the blood group that are infected with different diseases, right? For most diseases, it's roughly one third, one third, one third, right? But for cholera, 57% of people had the O blood group and then fewer with the other blood groups because there's some genes associated with the O antigen in the blood group that makes you more susceptible to get cholera, right? So much so that when you look actually at population patterns, that in the Bienges Delta, the vast majority of people, the O blood group is the rarest because you have a very strong disadvantage for the blood group. So it actually shaped the genetics of the entire population of the Ganges Delta. So the so bacteria can have a massive impact on human population. They can even shape the genetic landscape on who survives. This is this enormous selective pressure, and we don't usually think of these things, right? For human population, but these diseases can have that much impact, right? So because hundreds of years ago people started drinking brackish water, this actually changed the whole genetic landscape of an entire area, one of the most densely populated areas in the world, right? And it's not all lineage of Vibrio cholera that are pathogenic, right? It's just one small lineage because this bacteria is everywhere in water that are temperate, tropical, subtropical, equatorial. They are everywhere in the seawater, right? But there's this one lineage that likely evolved in the Ganges Delta that became adapted to humans, right? Because of our exposure to that brackish water. And it has this life cycle, right? It goes through the human gut because you swallow it in the water. It can be attached to zooplankton or phytoplankton, and you drink the water, and then you get sick, You get diarrhea and it goes back into the environment if you don't have a proper sanitation, right? So there's this cycle that can go on, and it selects for more pathogenic bacteria that can expand in the human host and go back to the environment. So you've got this human environment cycle that constantly is going on. Um, and to bring back the link to climate change, this is just a map of cholera cases, in the last 40 years or so, and the different epidemics that can be seen here. In Peru, there was one that happened in the 1990s that spread to the whole of South America. That was quite serious. We don't know where it come from. There's some hypothesis that the marine current would have brought this strain, but we don't really know. There was a huge outbreak in Haiti after the earthquake in 2010 because all the infrastructure was destroyed and people were homeless. So there was a massive, and this strain was actually brought by UN soldier that moved from Nepal, where they just had a cholera outbreak, and they brought the disease in the country. The UN took 5 years to admit it, and they never paid compensation. But that was also because of natural catastrophe. In Yemen, it was the war. I'm sure you've heard of that outbreak. Over 1 million cases, one of the biggest one in history. Um, uh, and then in 2022, there's actually a massive outbreak in Bangladesh. Okay, and more than 500,000 cases, which was not usual at all. Usually we get much fewer cases than this. And this is the first one I believe we can directly link to climate change. So if you look at the data from the ICDDRB hospital, which is in Dhaka, it's a research facility on the top floors, but the bottom floor is a hospital. And they count the number of patients that are coming in. Usually they get quite a number of patients. This is number of patients a day, so it's quite a lot. But during that summer 2022, it was much higher. And what happened in that summer is that it was much warmer than usual. The temperature didn't go below 32 degrees for months on end, and there was actually a drought. And both flood and drought can be contributing factor to waterborne diseases, right? The drought is because you don't have your normal source of water, you go to more suspicious sources of water, and then you can get contaminated that way. So there was a massive increase. So the climate change that changes the rain pattern leads to longer drought seasons and more intense short rain season that lead to flood, really increase the amount of these waterborne diseases, especially in coastal areas. There's weird things that happen as well. Just before I left Canada, this thing happened in Vancouver that was totally unheard of. So basically, um, there are some native communities that live on Vancouver Island, that's the west coast of Canada, and they have this traditional fishing that they do of airing eggs. So airing is a small fish, but they all spawn at the same time in the water. So basically, the water becomes thick with their eggs. So what the native people do, they put tree branches in there, and the eggs just stick to them, and they just pull them out, and then they eat them like caviar, right, raw. Um, but this is a problem. So in 2018, for the first time, they started getting people showing up with symptoms that it was not normal diarrhea. This was like cholera-like diarrhea. And this is completely unheard of, on the coast of Canada. So they eventually went back to the water, sampled the eggs, and they actually identified it was a Vibrio cholera, not the pandemic type, but a strain of Vibrio cholera that could cause these really drastic symptoms. But this had never been heard for before. But it's quite clear you've probably heard of the water blob that happened on the west coast um, uh, of America. There was a marine heat wave event. So there's a lot of these events that are becoming more frequent. So it probably is contributing to seeing weird things like this that were never there before. So we got to be prepared for more of these waterborne disease outbreak happening because of the climate change and the warming in the waters. Um, so I just want to talk a little bit about working in these coastal areas. So I've been working in Bangladesh for quite a long time. Um, it was my PhD student Tarek back in Canada, but he moved back to Bangladesh to run this project. Um, i- we're looking at waterborne diseases in coastal community, right? Because basically the whole of Bangladesh is basically a delta full of rivers everywhere, right? And it's fed by the Ganges, Brahmaputra, right? There's massive amount of fresh water then coming in contact with salt water. Um, and our project was basically so just to give you a bit of a location, Dhaka, the big city, is over here. And then you've got the Ganges that come here. Um, Madhbari is located here, our study site, and this is the Sundarban forest, which is the biggest mangrove forest in the world, right? Um, and so this is just a zoom in on this area of the village of Madbaria here. And you can see how this land is very developed, right, agriculturally. And this is a Sundarban. There's like zero development. There's multiple reasons. Well, for that is both tigers and also her typhoons that come through and is very, very dangerous. So that's why it's not built up, and now it's a protected area. Um, so the project was basically to look like how drinking natural water, sometimes brackish, right, is impacting the gut microbiome and your propensity to carry diseases, right? So we have two different households, households that were the control one that we gave them a water tank, a rainwater tank at the end of the study, and then the intervention where we gave them the water tank at the beginning of the study. And then we basically followed up what was happening in their gut and how often they were getting sick. So we picked mostly families with children that were quite vulnerable in different areas of Madbaria. And then we tracked their gut microbiome. Now we haven't finished the study, but we sequenced a few of their gut microbiome just to have an idea of what was going on of people that tested positive for different pathogens, just with a quick diagnostic test. And what we found out when we actually sequenced the gut microbiome was 15 people, is that a lot of them, none of them had cholera or symptoms. They didn't report any illness. But some of them were actually colonized with Vibrio cholerae, some of them with Vibrio fluvialis, and some of them with 3 different species of Vibrios even. Others were carrying other types of enteric pathogens, like Acinetobacter baumannii, E. coli. Almost everybody has that, but not necessarily the pathogenic type. Klebsiella pneumoniae, which was actually quite a high rate of colonization. In population like Singapore, it's mostly 10%. It was 95% over there. That causes a lot of diseases also. So, and we discovered that about 10% of the population was carrying Vibrio cholerae in their gut, although they weren't sick. So this is not something we're necessarily expecting. It's like these people don't have cholera. Why are they carrying these different waterborne pathogens, that like either salty or brackish water in their gut? And we're trying to figure this out, um, and whether it's specific strains that are becoming adapted to the human gut, or whether it's just a normal thing that when you drink water from natural sources, you got more chances of being colonized by these bacteria. They can actually live in your gut like as a part of your flora for a certain amount of time. But obviously, the environment as is having an impact on their gut flora. There's things that are not supposed to be there that are in their gut microbiota, right? Including some marine pathogens that we consider that if you detect this, you have a serious. I would come here to the hospital. They would detect this on a panel. They would send me for treatment right away. But there, it seems that it's regularly carried in a large proportion of the population. Okay, so we're trying to investigate that. Obviously, living and drinking natural water, living in a delta like this has an impact on your gut microbiome and your exposure to potentially pathogenic microbe and potentially to the evolution of novel pathogenic microbe, because the phenomena I described to you circulating through the human gut is selecting for more pathogenic organisms, right? So we're following this up. Another project we're doing is that this is my barrier over here, that we took a cruise in the Sundarban forest on a ship, that we went down from the top all the way down closer to the ocean, so that we could sample across the salinity gradient, because these bacteria are not only sensitive to temperature but also to the amount of salt in the water, right? So that way we could track the change in the different species along the salinity gradient. And this is something that was mentioned before in terms of remote sensing. Remote sensing data, you can tell salinity from satellite, but the resolution is not very good. You can also tell temperature from satellite, but you got much better resolution when you got ground truth information, right? So this is another thing that we need with Marine Station: collect more of that ground truth information to calibrate that satellite data, which if it's not calibrated, it is not that useful. And all the models you see published based on this cannot give you very accurate calls in terms of risk modeling if you don't have accurate data. So we're trying to establish a tighter correlation between salinity and presence of different pathogenic species by looking at that information and going through these salt gradients. So we did find some. We haven't analyzed all the data, but it was a clear different rivers. You can see that have different the high salinity versus the low salinity, and you can see that the species makeup changed. Vibrio cholera, it likes the less salty waters, but the other pathogens like Fluvialis, Alginolicus, they prefer the salty water, although Fluvialis says it can tolerate lower salinity. So you can see the ecology here of these different microbes, these different pathogens being influenced by the environment, and that if we can gather more information on that, we're going to have much better risk modeling, right? Um, so this is my colleagues, Andrew Kimberly from Tufts University. Munir Alam is my long-term colleague at ICDDRB. They look like they're not working, and That's actually true. Uh, Kim is 7 months pregnant, so that's okay. She gets a pass. But you can see I'm working downstairs. I'm filtering some water, and I'm not really drinking the water over there. Um, so, um, and one of the reasons why the Sundarban is not a place where people live is because there's a lot of tigers. Like, so only those with strong belief in rebirth should risk going near, right? So we had an armed guard with us the whole time, but I'm not sure, if he would have run or shot the tiger. I'm not sure. So we did take a water sample from inside a tiger paw in there. They can swim very well, by the way, tigers. So you have to be very careful with that. Um, so let me just check on time here. I haven't had my 10-minute warning yet. What is the here? I'm good. Okay, okay. So let me talk a little bit about some of the work we've been doing over here as well on St. John's Island. Okay. So this so we started a project to monitor pathogens in the local waters. And now I'm not talking about just regular marine bacteria. I'm talking about also human bacteria that contaminate the seawater, right? And if you look at the top 10 bacteria killers, you can see this nice list here. It includes E. coli, Staphylococcus, Klebsiella, some of them I mentioned before, Acinetobacter, Enterobacter. So these are all enteric pathogens or skin bacteria that are associated with humans, and they're some of the biggest killers in the world population. So they're called the escape bacteria. I call them the SKPs. So that all the main ones are included here. So these are the one we were targeting in the water because we wanted to understand better. They're very hard to detect in the environment because they're in low concentration, but that doesn't mean they're not important because they can get spread around by currents and going around different areas. It's important to know whether they're present in the seawater and even surviving in the seawater, um, and also to look at the risk it poses for recreational purposes, right? Um, so we were trying to survey different environment around St. John Island. We're including humans too, so we're sampling people that do some diving, as well as people that don't do diving, see if they harbor more of these antimicrobial resistant microbe in their gut. We're working on wastewater, on ocean water, also what's associated with mostly with seaweeds and also the sediments. So just looking if there's some reservoir for these bacteria in the environment. So the technique we use actually is that we actually enrich for the bacteria, right? Um, so the traditional way to test for water quality, right? You put some water on the petri dish and you count your E. coli colonies, but that's very time consuming. So what we do, we actually sweep the entire plate and sequence everything at once to know all the species that are there. And this is called culture enriched metagenomics. And we use this because the concentration are very low in these waters, very difficult to detect them. So, but if you enrich them, then you can get more of an idea of what's in the water. Um, so just to give you an overview of how the enrichment works, if you just sequence the seawater, right, and marine microbial ecologists in the audience, they're going to appreciate these are typical marine bacteria, Pseudothermomonas. Theobacter is Rebecca's favorite, is in there. So the top 10 are all things, they don't cause disease. But if you enrich, your top bacteria become very different, right? In the raw water, you have very, very few of these pathogens. But if you enrich, of course, you get a lot of these pathogens, right? So this is a very good way to get a look at these pathogens that are in very low concentration in the environment, but see if they're present and what they look like, right? And if you actually look at the top 15 organisms that are enriched, the Vibrios are at the top because they grow very fast, but they're also important to keep track of. But all these human enteric pathogen in red, they're in the top 15. So you can capture almost all of them by just enriching the water. Okay. So that's it's a very convenient way to just have a look. Oh, do I have these human pathogen contaminating my aquatic environment here? And if you break it down in the Vibrios, you can see that in Singapore, we actually had a lot of Vibrio fluvialis that was growing, right? So we're investigating if this is an ecological reason, but that's also one of the top disease causing in Singapore. So maybe this is something to do with it as well, right? But you find most of the pathogens in the water here. Um, so one of the things I love this quote from Prof. Bruce Levin that says that basically to pathogenic microparasite, we're just little more than soft, thin-walled flasks of culture media, right? Because we look from a human perspective, but from the bacteria's perspective, we're just juicy little pieces of growth media, right? And this is actually important when you do cultivation because when you enrich, right, you're giving them nutrients. So this is basically like, the nutrients is mimicking what's in your body. They like to grow on this. So it actually strongly favors pathogens because they're the one that grow fast because they're the one that could exploit our body, which is a nutrient flask, right? So if you look at the top ones that we've isolated from there, most of them are pathogens, right? A few of them are novel species we've never seen before, but you got these Klebsiella pneumoniae, E. coli, the Verbifluvialis I was talking about, the Paramolyticus. So you get all these pathogens because the pathogens, they're pathogens because they can quickly exploit rich nutrient sources, um, which can be media or the human body, right? Um, and this one of these ones that we've found, this Vibrio naceae 76, is very abundant, but it had never been seen before. So we were trying to find a new species name for this one, and I don't know if you guys know much about the history of different islands, but if you look at Sentosa Island, right? This is the touristy island with Universal Studios and everything on it, right? It used to actually be called Pulau Belakang Mati, and which means island behind which lies death. And you talk to different people, depending on how old they are, they have a different story of why it's called like this. It's because of pirates, because of British cannons, it's because of infectious diseases. So I don't know what's the truth, but that it had a bad rep back then. But it was renamed Tranquility Island for the tourists, right? So if it's non-pathogenic, we'll call it Centosia, but if it's pathogenic, we'll call it Black Maria. Um, so you can also select for things that you want when you're doing enrichment. So if you put antibiotics in the media, you can see a lot of the species. These are all the pathogenic species in red. A lot of the species drop out, right? And E. coli and Klebsiella are the one that survive the antibiotics, which makes sense because they're the one most often carrying antibiotic resistance. So you can also directly target the one that are most worrisome in terms of infection. Um, Aeromonas is actually it's actually a natural, aquatic bacteria, but it also people have hypothesized that it can be a reservoir for antibiotic resistance because it's one of the few ones that can survive. So this is something to be looked at for sure. Um, so our goal by doing this is actually to do what we call preemptive epidemiology. So epidemiology, you've all heard about that. You got an outbreak, you've got epidemiologists, they go in, they try to trace, contact tracing, how it got passed around, the source, etc. But a lot of the time, the problem is that you don't have enough data to know where it come from. So preemptive epidemiology is to actually gather data on what's circulating in the background in the population. So you can take stool sample, wastewater sample, but also, aquatic sample to see what's in the marine environment as well. And then you get that epidemiological information. Oops, before the outbreak actually happens. And this is all our strains we isolated from the water in Singapore. They're all Klebsiella pneumoniae. The one in green we selected in a traditional way on a culture media and picking colonies. The other one comes from our sweeping the plates and just sequencing everything. And then this is a referencing. So you can see that there's different sequence types that are found across the world in different countries that have been linked to different types of outbreak in hospitals or in the population. So if we gather enough of that information, we're going to know the landscape of pathogens in our environment. And when there's infections, we're going to be able to tell where it came from and maybe link it to a cause. So this is why I think it's important to have this prospective data on these pathogens, right? And one of the things I want to mention here, it's that even here at the equator, the temperature is rising. So this is data around Malaysia and Singapore, right? But I love this quote by Arturo Casadevall, who's the president of the American Clinical Pharmacology Society. And he says, people say the world's only warming by 1 degree, right? It's not a big deal. But that's not the right way to think about it. For microorganisms, every day that is hot, every marine heat wave is a selection event, just like antibiotics, right? You take antibiotics, only antibiotic resistant microbes survive, right? Well, there's a heat wave, only the heat resistant microbes survive, right? So this is not good because our body temperature is 37 degrees. So the more they adapt to heat, the more they're able to infect us, right? So I was intending to finish there, but then I heard Minister Lee talk about oil spill. So I wanted to talk a little bit about oil spill. Do I have time to talk about that? Yeah. Okay. Good. Good. So he mentioned, I think Yanni put a bit in a big application to look at, and I think that was a very positive signal from the minister that maybe this was going to be funded. But we started sampling, so I don't know about politics here, if that was a promise or you know. Um, but anyways, it makes me feel positive about it because we collected all those samples and we need to be able to analyze them. So we actually jumped on it as soon as I actually remember getting a call from Rebecca, my wife, and in the middle of the night, and she says I was in America at the time in a conference, and she says there's been no space in Singapore. You're gonna get your student to do drone sampling now. I'm like, it's 2 in the morning. What is happening? But she was right. So she drove this effort for us to just get right away and get sampling, because if you want to know what's going on ecologically, you got to get sampling as soon as the oil spill happened, right? So Minister Lee has already described this, and it affected actually quite a large zone, including St. John's Island, and there was a lot of oil residues on the beach, right? So what the team did is that people from South Sea, so this is the Singapore Center for Environmental Life Science Engineering, that's even worse than SGINML as an acronym. But this is the center both Rebecca and I work for. It's located at NTU and NUS. It's a multi-university center. Um, and we're mostly focused on microbes in the environment. So we basically focused on sampling around St. John's Island and doing a long time series. And different people have different expertise in South Sea. So Janelle was trying to isolate some oil degrading bacteria for bioremediation. So this is another example of difference in ecology, right? The strains they use in the UK or the US for bioremediation, they're probably not very good here because the water temperature is too hot. So other bacteria would do a lot better. So we got to get our own oil degrading bacteria. So that's what we're trying to do. Hailim is looking at the effect on turtles that are nesting on beaches on the East Coast Park and different places in Singapore. And I was more doing drone sampling. Um, so there's these 3 sites you're gonna see tomorrow when you go to St. John Island. You go to some of these sites. They've been cleaned up now. Okay, but the Inias and Buoy was one of them. Bandery Bay and also Eagle Bay was the ones that we're still sampling. So this is Precious is sitting somewhere in the audience. She is not Swedish. She is Singaporean. But our boss was Swedish, and I noticed that her outfit was very Swedish. So I pointed that out to her. But yeah, so she was sampling right on the shore. So you can see how the seawalls were completely covered with oil, and it is amazing the cleanup job that they have done here. Because I went back for like staying overnight on St. John's, and these walls had all been scrubbed with pressure hoses. I mean, it was kilometers of walls. They did an amazing job at cleaning this up. Um, but we wanted to see the effect throughout the cleanup. You can see the water here, you can see the oil floating around, and you can see that it had penetrated in the sediments at that time before they shoveled it all up, right? So we wanted to see how much this is impacting the microbial population and if some oil degrading strains are being enriched there, right? And here it's like Minister Lee was saying, we are in a hot spot for oil spills, right? There's almost every 2 years is some kind of oil spill over 34 times that's happening around in the area. So it's important to understand ecological impact, not just that oil spill, but in general, and also how also the microbes are reacting to it. They play because they play a role in the cleanup, not just us. They also play a role in the cleanup. So we went up to sequence. We get our things together. Not me because I was in the United States, but 2 days after we started sampling, and Prasha and Clarence that are here, they went every 3 days. It's an amazing effort on their part, and then they switched to every week, and now every month. So we'll finish around just before Christmas this sampling regime. This is just a little video. Can we push play on this for the program? Yeah. So this is the drone sampling we're doing. It's very simple, right? It's just a drone that is waterproof, and we put water bottles on it for sampling, and we just basically just dump it in the water and then bring back the water. And you can see here how oily the water is, right? So the first couple of days, it wasn't really safe because of the smell and everything to go in, but you could send the drone in the water and safely take some water sample. And you can also go further, so you can compare um, more far shore to near shore, how much they're impacted by the oil, right? Um, and then what we do after that with the sample is that basically we filter the water, and then we can look at the microbial community, but also eukaryotic community, eukaryotic DNA possibly in this. But we can also cultivate some bacteria to see the impact on the microbial population, and hopefully, like Janelle is doing, isolate some oil degrading microbes from those same samples, right? So let's just see. Okay, so that was that my last slide? Okay, was my last slide. Sorry. So we've got ongoing efforts, and hopefully we'll be able to have a look and maybe get some new clues on how it's impacting the microbes, but also the eukaryotes in the environment. Okay. So yeah, so that was just giving you a bit of an overview, and I think it's a fantastic gathering here. It's so interesting to see people from with similar interests from different parts of Southeast Asia, and I'm really looking forward to talking to all of you. Thank you very much.
Thank you, Prof. Boucher. We do have a bit of time for some questions. If
Okay.
anyone in the audience has any questions, maybe let me pass you the microphone.
I'm not sure how is it in Singapore, but like for us, I noticed that if there's very heavy rainfall, it changes the temperature quite drastically in the drinking waters. And in the case of aquaculture farms, they even have crashes in the fish populations in the forest after the rains. I want
Oh.
to kind of look at that. Since yesterday they shared that there's quite a few aquaculture sites around Singapore. If you had a chance to look at the Vibrio and the populations around the aquaculture farms.
No, so far I haven't done any work with aquaculture. But it's certainly I know that there's some work going on with aquaculture here, but we haven't tracked that down. But that's a very good suggestion to have a look at that because they both can be the victims of the pathogen, but they can also contribute to the increase in pathogen with the nutrient output from the farm. So yeah, but we haven't looked at that. But yeah, it's a great suggestion to do that. Yeah.
Question. So during to focus your areas of
So I didn't show that, but we worked a little bit with Marine Ports Authority of Singapore, and there's some great modelers that are doing some work on that. And Prasha actually went on the boat with the MPA, and they went to sample the sediments and the water around where the oil spill happened and where the slick was most present. This was 2 weeks after the event, just for logistical reasons, right? We didn't get the MP didn't organize the cruise until then. But yes, we did, and we're actually working on analyzing the amount of oil in these samples, and we managed to get also a neat oil sample from the actual tanker from MPA. So hopefully we can put all that together in time and get a better idea. Yeah.
and
Okay. Yeah. So yeah.
like a month after
Oh wow. Okay. Yeah. Yeah.
you know identify.
Yeah.
So maybe if I could that.
Do you have info on that, Rebecca? Or?
Side. So where which all been studies, so like under experiment studies, so we've been sample there, we've been sampling Eagle Bay with the sediments before, and the extent is obviously like serious. So there, that's actually why I'm sure is that yeah, I was talking a little bit of where we took oil samples. So when we're taking environmental where thinking
Because you know I don't see the what the oil there.
Yeah.
so when to because
In terms of drinking water, this is the purview of PUB, right? And they said that it was safe, they didn't get contaminated. But I haven't, been working with them. They do their own testing, their own thing, because we have a lot of reservoirs in Singapore. Basically, every single river is blocked so we can keep the rainwater on the island for drinking water, right? Um, but hopefully, I think they have buoy system that they block so it doesn't come anywhere close. Hopefully, so as far as I know, there hasn't been any contamination. But that really depends on the setup that you have, right? But this is definitely a concern. If your drinking water is in contact with the ocean, somehow, yeah, there's a real risk of contamination. It can be pretty serious if it gets into the drinking water. Yeah. Yeah. No, it's the people.
smell great. We actually have advisories. When you live in California, not to drink tap water because of this infection. How they've got active water.
Yeah.
Great, well, thanks so much. I learned a lot about microbes today. So I've got a question like from a forecasting point of view. Is there like any work that's or an interest in kind of because like you can forecast like when like it's gonna fires and
Glenn Parks give money to Patrick Martin. I'm on his grant. Then we will find out about this because so Patrick has this long-term time series is about geochemistry and nutrients, right, of the waters around Singapore, and is working with some modelers. But talking to him, I realized, oh, it would be great to add some microbiological data into this. So we're trying to team up and to do this. And so yeah, we're intending to look at this. There's also a lot of interesting correlations. For example, algal blooms, right? We know they're becoming more frequent around the world. There's some really nice satellite work that's been done. We're planning, Rebecca and I, to work on this to look at the association between bacterial pathogen and algal blooms because some algae contain a lot of chitins and Vibrios love chitin. So we're trying to see if there's an impact on the amount of pathogens when there's an algal bloom, if it correlates, so if there's effects like this. And that could come into play into modeling. Also, in terms of modeling, there's a lot of really high impact paper that talk about the risk of vibriosis on the coast and how it's going to go in the future with climate change. But they're only as good as the data they have from the ground as well, because salinity and temperature are two determining factors. And satellites are not great at determining salinity, as I'm sure you know. So we need a lot more ground truthing data. So we're trying to work on this to try to improve the models to gather more. So how do the microbes correlate with the environmental parameters? And then how can we improve the satellites, measurements of these parameters to maybe fold that into bigger scale models? Yeah. For cholera and vibrosis, some models have been developed. There's a few ones that have been published that are good at predicting when outbreaks going to happen, and they take into account both water parameters but also social parameters as well. So there's some work in this, but I think these models are in an early stage. They a little bit. Actually, there's a few countries. There's some test cases. Rito Caldwell's team in the U.S. has worked with several countries in Africa and in Bangladesh to predict cholera outbreak, and they've actually implemented vaccination strategy based on this. But now we have a global cholera vaccine shortage, so that's not very helpful. Yeah. Yeah. No, I mean, one of them is we're already doing it with E. coli on the beach, right? It's like a don't go swimming right now. It's risky, right? So there could be some advisory type of thing, right? Make people more aware. If you have a cut on your skin or a burn, don't go in the water, right? So there's education as well. But yeah, in terms of the prediction, it's hard, but I think it's mixed with education, right? Like for example, hurricanes, like what's happening right now, you can inform people like, look, when hurricanes happen, there's also this risk of infectious disease. So just be conscious of it, right? So I mean, the prediction of vibrio in the modeling, yeah, it's true. It's only useful as we can create intervention to tell us about it. Otherwise, it's just making people feel scared, right? But I do think there's some advisory and things like that can be done for that. For cholera, there's vaccination campaign is possible. The other ones, there's no such thing. But advisory, don't eat the seafood. This type we're already doing for HABs, things like that. Bring that into the implementation. That's the way I see that can be done, right?
I think that'd be really
Yeah.
important because often, like near the coast, I mean, I live right near the coast,
Yeah.
like Cornwall, UK, and locals will know things like if it's heavy rain and there's a river going into the sea, you don't swim.
Yeah.
The locals know that.
There's the famous UK study called the Beach Bum Survey that they took surfers in the UK. I don't know what people do surf in the UK, but apparently they do. It's too freezing cold, but they had like much higher AMR E. coli in their gut, the surfers compared to normal people because of sewage outflow when it's raining too much, right? And this is what inspired my study that I'm doing, asking all the divers and the swimmers here to see. I want to see if there's the same effect here. But yeah, yeah, that's an important consideration. It's true. Yeah. Yeah.
Once again, that's all the Q&A segment. So thank you so much once again, Prof. Boucher. Maybe I could just invite Steve Widdicombe from the Plymouth Marine Laboratory. So that's all I mentioned. Widdicombe, we met yesterday, is an ecologist with over 10 years experience in using fuels international level of scientists working on the drivers and impacts of ocean acidification. Prof. Findlay, who I believe will be speaking first, is a biological oceanographer and senior scientist. Her research focuses on the effects of climate change and ocean acidification on marine ecosystems and how to apply this knowledge to maintain a healthy, sustainable ocean. Professor Findlay and Professor Widdicombe, please. Can you pass this to you? Thank you so much.
Thank you. Thank you very much. It's lovely to speak to you all here today, and thank you, Yanni and the team, for organising this event and inviting me and Steve and the rest of us along from the UK and the British Commission for supporting that. So I'm going to give you a kind of overview of ocean acidification from a global perspective, thinking then down into the local perspective, and then hand over to Steve to talk more about the biological aspects and biological chemical monitoring, I believe, possibly. So this is just to start off the ocean acidification stripes. So many of you maybe have heard of the climate warming stripes. They were created by Ed Hawkins in the UK a number of years ago now, and they've been quite a powerful worldwide communicating tool for climate change. From one of the projects that we've been involved in that's using attempting to kind of use satellite data to predict ocean acidification parameters around the world, we've now created these ocean acidification stripes, and you can go onto the website and have a look, and you can download regional values for your regional stripes for your region of both pH and aragonite saturation state. So just to highlight those ones. So this is actually global decline in acidification over the last 30, 40 years, and it's declined by about 27%. So what are we talking about when we talk about ocean acidification? I'm not going to go into this a lot because I think we've got Patrick talking about carbonate chemistry tomorrow. But just so kind of everyone's on the same page, this is the chemistry of the ocean. So what we have is, you'll have heard some of these terms mentioned yesterday, total alkalinity, and many of you are probably familiar with them, pH and saturation state. So we often talk about CO2, we often hear about hydrogen ions or pH, and this saturation state is also quite important when we're thinking about ocean acidification. And what's happening with ocean acidification is basically we're adding lots of CO2 into the atmosphere that's rapidly going up into the oceans. It naturally dissociates, it naturally combines with water and then dissociates into these bicarbonate hydrogen ions. And within this system, you kind of get a measure of the pH and a measure of the carbonate ions. So I won't go into detail, but basically what we're seeing with ocean acidification is you add more carbon dioxide, you get a decrease in the acidification, so an increase in hydrogen ions and a decrease in these carbonate ions here, which is a decrease in the saturation state of really important minerals, calcium carbonate minerals, aragonite and calcite, which I'm sure Steve will mention more of. So what is going on in a global perspective? So these are models these are the CMIP6 models, so the latest models that are used in the IPCC reports that just show how we've changed from the 1850 pre-industrial time in terms of pH to present day. We're around about here now, which is where the models start to diverge. As we go forward in time, we're going to really be seeing which route we go down over the next couple of years, but it's around mid-2020s that actually we start to see the models diverge. So we've seen about a 40% decline in pH since pre-industrial times to present day. And this is the same for aragonite saturation state. So you can see we've kind of declined now to around about 2.75 in a global perspective, global mean value of aragonite saturation state. And yeah, going forward, you can see depending on which scenario we choose, so the kind of worst case emission scenarios, these middle emission scenarios and the 1.5 degree Paris Agreement scenarios will really depend where we end up in terms of acidification, in terms of pH, but also things like the saturation state. So what does the regional variability look? So we talk a lot about global averages, but it's the same thing with temperature. We have this global 1.5 or 1 degree warming, but actually on a regional scale, they vary hugely across the oceans. So this is again pH on the left here and aragonite saturation state on the right from the same set of models. So this is the model ensemble of those climate change, the CMIP6 models. And you can see that there is a large amount of variability across the ocean, both in pH, but actually really importantly, aragonite saturation state. So the polar regions have a huge amount of fresh water input, particularly in the Arctic, and that fresh water naturally means that there's less carbonate ions in the system, and so you actually get lower aragonite saturation state and much less ability to buffer changes in acidification in these regions where you get high freshwater influence. And it's really important to make sure that we consider these because you hear a lot about these the planetary health check. I don't know how many people are aware of that, has just come out, which has these 9 planetary boundaries, which we've just crossed. And according to that, we haven't quite made it across the boundary for acidification yet. But if you took into account all of this regional variability, and I'll show you a little bit of that in a minute, is that actually many of these places have crossed that boundary if you took that into account. So this is some of the work that we're trying to do to kind of make an assessment of that acidification boundary. It's a little bit complicated, but I'll take you through each step. So on the top row is pre-industrial, the year 2020 minus pre-industrial. So the change between present day and what was going on in the pre-industrial. And the color bar is the percentage change in time in that time period. The next row is between 2050 and pre-industrial, and the final bottom row is what happens between the year 2100 and pre-industrial. And each column marks a different emissions scenario. So the first column is low emissions, then you have a mid emissions, and then a high emissions scenario on the far right. So what you can see that at present day, many of the oceans in the tropical areas have seen a decline in aragonite saturation state by between 10 and 20%. But as you move into these polar regions, and there's not much difference in 2020s between the 3 scenarios, but you see that actually some of these are seeing nearly 30% change since pre-industrial conditions. As we move forward in time, that change in the polar regions is much more obvious. We're seeing maybe 50% change from pre-industrial, whereas in the tropical regions, actually, maybe we're still only 20–30% change. And again, as you go into the future, depending on which scenario you choose or which scenario we end up following, you see quite big differences then in the sort of amount of change we've got since pre-industrial conditions. So in the worst case, kind of high emission scenarios, you're seeing up to 60 to 70, even 80% change since the pre-industrial levels. Whereas actually in the low emission scenario, this is an extreme low emission scenario, so one we're unlikely to follow. But actually we start heading backwards. We start removing CO2 from the atmosphere, and we're starting to able to actually see some improvement and move towards back towards the pre-industrial state. So that's what the models tell us. And obviously those models are, like we've heard many times, only as good as the data that we have to support them and validate them. So this is the presentation, an older version, but that's fine. So this is the Global Ocean Acidification Observing Network Data Explorer. So this is a a portal that you can go on to see where all the acidification observing network observing activities are taking place across the globe that have obviously been registered. And within Go On, what we try and do is we have regional hubs, and we try and actually audit what's going on in terms of activities. And so many of these are individual stations. You've got the Argo floats on here as well. You've got so bioargo. So Argo has temperature and salinity, but you have bioargo, which actually also has pH measurements on them as well. And then you've got some of the cruises that are going up. And you can see there's an interesting sparsity, perhaps, in the Indian sector, Indian Ocean sector. We've got some sites on here. I would urge people, if you're monitoring pH or acidification parameters, to just check and see about registering, to go on more broadly, but actually thinking about registering your sites onto the Go On data portal. It's very easy to do so. And then we can kind of capture that network. It's not, you don't enter your data, it's just a metadata. You can go on and click on the station and see who's working in that region, what's being monitored. So from those kind of those data observations, what can we understand about the regional variability? So I've kind of gone through some different ways that we can think about the data sets. So these are examples of long-term time series. And then in a new version, which I haven't managed to get to you in time, obviously, I also put some of the updated European regional versions, but don't worry, it's my fault for sending it late last night. So, but yeah, so what we have here is the longest time series we have for carbonate chemistry. So the Hawaiian Ocean time series, Bermuda Atlantic time series, and Canary Islands, which is ESTOC. And all of those show roughly a similar rate of change in pH. So this is PCO2 in the oceans, and then the more lower CO2 in black there for the atmospheric CO2. And then this is the carbonate ion equivalent to the kind of saturation state. And they all show very similar rates of decline, which is kind of what the global average is telling us. It's about 0.017 pH unit per year, I think. But actually, if you start to look at other regional, although slightly shorter time series, you start to get much more variability in range. And across Europe, we're seeing, as you go towards the coastal regions, you get more rapid acidification events happening, which becomes more complicated. So some of the other things that we can start to do with that data is to start to put them together to produce products. And we've heard about SOCAT and GLODAP, I think, already. But these are just two of the data products that are collecting all those individual data and putting them together to look at what's going on a global and regional perspective. So this is, again, kind of SOCAT data, so the Surface Ocean CO2 Atlas, and then GLODAP is the Global Ocean Data Analysis Project. And this is pH from both of those products mapped onto a grid. So you can see, again, that regional variability in terms of the data. You've got high pH in the tropics, and then you've got these spots of where the fresh water comes in, and actually you've got different dynamics going on. It's high pH because it's cold, so this isn't temperature corrected pH. But also from you can see the surface water CO2 values and how the number of data points we've got have increased through time. So in the 50s and 60s, there was a few transects that were collecting CO2, a few more in the 70s. And you can see that as we've gone through time, we've actually increased the amount of CO2 data available. But actually, if you put that together, you can see just how much the CO2 has kind of shifted as we go along from the 70s, 80s, 90s, 2000s, and that's 2000 to 2010 to 2015, 14. So the atmospheric CO2 in that period was 354. We're now at 420, I think, in the atmosphere. So again, if we were to update this, it would probably be more like over this period here. What's interesting is that actually we're seeing, if we were to add on the latest data, so the 2020s, we're actually not necessarily having an increase in CO2 measurements anymore. We've kind of almost plateaued, and that's I think Matt mentioned it, or Steve yesterday, where we're actually seeing less time series and less shutting down of many of these programs, and they're at risk all the time of actually less data becoming available. And then we can also look more about some of these. Nowadays, we're seeing more of these kind of match-up machine learning type of products. So I just wanted to mention a couple of them that we've been working on and some that our colleagues have. So we've got things like the Ocean Soda Matchups Database, which is a European Space Agency project, which has taken a gridded data approach to collate all of the available carbonate chemistry data that we have, both from in situ observations, but also from satellite products, so be able to match that up. And that's really a tool that's available now to download for model verification, but also for making some of these additional assessments over regional and global scales. As part of that project also, there's the Ocean Soda ETH-Z product, which is a machine learning but data-driven product. So again, it uses a combination of in situ and satellite observations to predict the carbonate chemistry. And you can see the rate of change in the saturation state here between 1980 and the present day, and the rate of change in pH at the bottom here. So the rate of change for aragonite saturation state based on this data observation suggests that actually in the tropical regions, you're getting the fastest rates of change, whereas for pH, you're getting the fastest rates of change in the polar regions. And that's because of this freshwater buffering ability that the polar oceans aren't able to buffer the change. And so actually the observation is telling us that actually depending on where you are in the region, in the ocean, the rates of change are actually different. And these products are now available through something called the Ocean Data Lab portal. So this is the address for it, acidification.oceandatalab.com. And you can go on and you can zoom into any location around the world, and you can choose carbon dioxide, you can look at pH, you can use aragonite saturation state. And the products are available for a 1/4 degree, 8 daily resolution. So relatively fine scale resolution, considering the kind of the scale of these types of models. And then there's the equivalent from the Copernicus group. So again, they've got similar products that are machine learning data driven products. And there's a data link there to have a look at those, and they actually have forecasting products as well, so you can actually look at the forecasting of some of those products. But again, that's all data driven, so it's only as good as the data behind it. And much of that uncertainty, you can see that in these plots are areas where actually we have low data. So for pH, much of the Arctic Ocean is still very data poor. And actually things like aragonite saturation state, you can see that there's highest uncertainty in the models, in these machine learning models for those regions in the tropics. And again, that's a kind of data poor regions. So do they match up, the models and the observations? So on a global scale, yes, they seem to do that. So this was work by Dick Feely and others that came out last year that showed that if you take these decadal trends and look at how they change through time, the gray bars are the observations, the blue and the red are different model scenarios, and they really match pretty closely in all these parameters. And as a result, you can kind of then use a combination of the models and the observations to just track how acidification has changed through time. So when we think about how variability manifests, we can start to understand just how complicated the system is. And so this was an OSPAR report that we wrote, it was published last year, and it was the first time acidification was considered in this quality assessment that happens in Europe for the Northeast Atlantic region. And the assessment happens roughly every 10 years. And acidification was included in it for the first time this year. So we talked a little bit about what was happening. We looked at the trends across the region, and also tried to understand and discuss the different aspects and processes that were driving the regional variability. So just on a kind of spatial scale versus a temporal scale, you can kind of start to see what activities, what drivers are changing carbonate chemistry at different scales. So on the kind of bigger scales, you've got water mass circulation properties will affect it. But as you move towards the sort of fine scale, in both time and space, you've got things like nutrient cycling, dissolution, methane oxygenation, and the kind of the biological processes, photosynthesis, respiration, will affect things on a sort of daily fine scale. And in the middle there, we kind of have like ship emissions, nutrient runoff from land, land interactions, stratification, freshwater input, things like that. So it gets really complicated, especially as you get nearer coastal systems. And so the pH, many people argue that we should have kind of this overarching ocean acidification, which is the CO2-driven impact of climate change. But actually in coastal regions, it's much more complicated. And what's happening in that that global perspective is actually either kind of impacting, enhancing kind of coastal acidification, or actually kind of ameliorating it in some ways. And we can look at that in a different way. This is a report we did just for the UK. So we looked at around the UK shelf seas, what is the surface CO2 parameters, and how they change through season and distance from land. So as you're really close to land, you have a huge variability in terms of the CO2 across different seasons. Whereas as you move offshore, you get much less variability. And that's because as you move towards land, you get much more additional CO2 from river mixing, from land influence, from remineralization processes. And in the summer months, you get much more primary production, which is driving the CO2 back down. So what does all that variability mean when we think about the impacts of that? So that's something else that we've been interested in, is kind of understanding that natural variability and trying to understand organism response in terms of what we see in the laboratory, but also what we're going to see moving forward in time. So this is some work that we've been doing off the back of this space agency project. So we've got a lot more of data. So we've been using this Ocean Seda time series data, which I showed you before. And looking at the Pacific oyster, I'm just going to give a couple of examples. So this is the Pacific oyster pH environmental envelope. So quite often we hear about temperature environmental envelopes, and people are quite familiar with that. But it's never really been done so well with the carbonate chemistry because we haven't had the data to be able to do it before. So what we did here is split the time series into the first 10 years of the time series, so the 1980s, and then the last 10 years of the time series is in red here, so between 2012 and 2022. And you can see that over time, the pH in those, if you take the data from the location that you find all these Pacific oysters, has shifted. We expect to see a decrease in pH, and we find that. So if you assume that these things haven't moved with time, then this is the pH envelopes that they will have experienced over that time period. And we can actually then subset that into the different regions. And so you've got places, Alaska, Australia, the East Australia, West Australia, California current here. This is the Northeast continental shelf. So this area here around us. You've got one site, I think, from the Indian East Ocean, and you've got a couple of sites around the Kurihori current. I can never say that properly. But what you can see is that actually all of these have different temperature envelope, pH envelopes, which is, it's kind of not surprising, but it's the first time we've really been able to map these on this kind of scale. So you can see that, for example, in the Northeast European shelf, or the Northeast continental shelf, they kind of, these bars here represent the mean of all of the data. So they sit kind of quite nicely where between the mean of the present day has shifted to the past. And it's very similar to this overall picture. But if you look at somewhere like the Tasmanian Sea, New Zealand, actually they were 40 years ago sitting kind of above that. And now they're actually in an area which is kind of more like the average, compared to maybe somewhere like the Californian current, which was kind of more average 40 years ago, but now is actually below average. So we can start to pick out how these systems are changing in terms of where they're experiencing different conditions and the environmental gradients. The other example is a bit older work. So this is work from the Arctic we did back in 2010-11. And what we were able to do was to go up into the Arctic and measure the carbonate chemistry through time, but also through depth profiles. And you can see that the black lines here represent the pH, present day pH, and this is estimated future pH conditions as you go from the surface down into the deep oceans, and CO2 conditions here. And what we did was, alongside of that carbonate chemistry measurements, we also took samples of different copepod species. So Calanus species, we did a period of 24-hour sampling to get the vertical profiles of these species. And we found that these copepods were, the adults were migrating across the water column across that whole 24-hour period, whereas the smaller species, these Orthiona species, never really went below about 50 meters, and the same for the Nauplii, the early life stages. And then we ran a series of experiments in the lab to see how they responded to changes in CO2. And as you can see, so we went from a sort of present day CO2 to a high level CO2 and an even higher one. And the adult Calanus, these Calanus that were exposed to all of these conditions, so quite a wide range of pH conditions, had very minimal survival. Had most survival. So there was no difference between the survival across these different treatments. Whereas the Orthiona and the two nuclei, so the nuclei of the Calanus and the nuclei of the Orthiona, both don't see that different pH gradient. They see a very minimal pH gradient and actually had much more significant responses in terms of survival. So again, it's that being able to kind of think about what we see in the lab and match that up to the variability that these organisms would naturally experience. And the final example I just leave on is one that's very recent. So this is now where we're sort of moving towards a modeling system. So this was a recent paper by Hoffman, Elizondo et al., and some colleagues of ours. Nina Bednarczyk was involved in this one. So this is pteropods. So these are tiny planktonic snails that float around. They've been a kind of indicator species for acidification, a kind of like polar bear for acidification. And what it shows is the dots indicate the different stages. So the different colors are the different stages of the pteropod's life. So yellow is eggs, blue larvae, red juveniles, and black are adults. And this is how they would migrate over day. This is where they would be located in day and night, in the springtime, in the summer, autumn, and winter. So you can see that we would expect to find lots of adults at depth in the day, in the spring, but maybe all of them in the surface waters at night. And the same, you've got kind of juveniles going up and down in the day and the nighttime. And it changes, obviously, when you've got different types of the larval species there and the egg species, the egg. And we can map that observation and these models onto the data that we have for something like aragonite saturation state. So the color that's behind this is the aragonite saturation state that they observe and then modeled over that period in the Californian current ecosystem. And so where the color ends, so you've got these sort of purpley colors, is limited to 1.5 aragonite saturation state, and that's known to, once you get below 1.5, it's known to impact the shells of these organisms. So you can see now that they're starting to, when they're in the surface waters, they're generally not impacted by that low aragonite saturation state. But as they migrate down, you can start to see how long they spend in that essentially detrimental conditions, water. And what they were able to do was kind of plot how much percentage loss of calcium carbonate they have seen through time, based on the kind of the natural variability, the overall trend, and then the sort of extreme events you expect. And then that's shown an increase in probability of mortality going through time as well. So we're able to start to really understand how that regional or local variability is important for the organisms that are living and experiencing those conditions. So I'm going to end just by saying, so why is it important to monitor OA locally? I'm sure most of you can appreciate this by this point of the workshop, but I show this to some of the policymakers we kind of work with locally. And it's really about, obviously we want to reduce the impact of acidification, and we can do that kind of by reducing emissions, but that's a very, like we've heard, national legislation is the way forward to actually be able to implement these international agreements. So there's things that we can do locally if we understand that local system that will actually allow us and allow organisms to be less impacted by acidification and reduce the impact. So we can manage co-stressors, things like land runoff, pollution, overfishing. We can invest in science and networks and monitoring. We can invest in adaptive planning, habitat protection. There's things that we're now looking at incorporating that local data into understanding how we can plan and manage the system. Education is hugely important, as I heard many times over the last day or so. And then even things like understanding kind of the seagrasses, how the blue carbon might help acidification. I think we hear a bit more about that on Thursday. And some of these other technologies, what are they going to do to reduce the impact of acidification at that local level? So just going to end there saying the chemistry is clear. Acidification is happening in many locations, but the regions of most rapid acidification depend on what you report. So if we're thinking about pH, that changes most rapidly in these polar regions. But actually, if we think about something like aragonite saturation state, then that seems to be most rapidly changing in the tropical regions. Variability matters when we think about investigating the impacts and understanding what those organisms have experienced historically, but also as they go forward into the future, extreme events are going to be really important. And handing over to Steve, more co-located observations and monitoring are needed to be able to make these better assessments. Thank you. Oh, yeah.
Okay, thanks very much. I'm conscious I'm the person between you and if you've got a question, I mean, whether we do the questions at the end, but
Our photo period is different, right? Our dynamic cycle. So do you think that's actually going to affect our weight more because we have much longer periods than we do in temperate environments?
I think what would be important is understanding what they do here locally. So if you can understand if they vertically migrate more or if they don't, then you can start to piece that together with the kind of the time series that you'll have that environmental data, and then put those two together, and you can actually start to piece together what they're experiencing. So it is probably different, which is why locally I think you would need to understand that local dynamics. Yeah.
And just because you're at PML, I have to ask you, have you done it with eHubs to see how it migrates?
We haven't at PML. MBA are really into eHugs, but we've got some collaborations that will start up with them to look at sort of NCDR side of eHugs impact. So we might look at the variability. Yeah. So.
did you do your masters on here, eHuts? Yeah. Right. Okay. Thank you very much. We got the clicker. Please. Did you? Ah, lovely. Thank you very much. I might wander around to try and keep everyone with me while we're waiting for lunch. Okay, right. Biological principle. Thank you very much, Helen, for great introduction on what is happening in terms of ocean acidification and importantly coastal acidification. If you're scared of ocean acidification, you should be absolutely terrified of coastal acidification. It is progressing at a rate that is quite upsetting, really. So what are the biological impacts of ocean acidification? So, as we've seen, ocean conditions are changing, but I think there's some things that we really need to take home when we think about the biological consequences. And after all, it's chemistry can change and it's all very interesting, but from a human perspective, the ecosystem services that we rely on are largely governed and provided to us by the biological system. Now, the first thing is carbon dioxide is a natural part of biological processes. It's been part of our planet for billions or millions of years. So organisms have evolved having carbon dioxide in their environment and hardwired that into their physiology. So effectively, there are mechanisms in our bodies as well. Our instinct to breathe is governed by levels of carbon dioxide in our blood. So if you start changing levels of carbon dioxide and the carbonate system, you effectively have physiological pathways that are hardwired straight into an organism's response, which we've seen with natural variability over normal cycles. That's not a bad thing. In fact, that plasticity is really important to be able for an organism to continue to function in a naturally variable system. But it's when it exceeds that natural variability, then it becomes an issue. We've also seen with measures of aragonite saturation state that Helen showed that the reliance of many marine species on calcium carbonate structures makes them highly vulnerable to changes in carbonate chemistry and reduced carbonate saturation states. The provision not just the health of them as individuals, but if you think about corals and oyster reefs, merle beds. You're talking not only about the loss of an individual species, but the loss of a whole biogenic habitat that takes with it all the associated biodiversity. Marine organisms actually incur an energetic cost in dealing with ocean acidification, and I'll explain a little bit more about that as we go through the talk. But effectively, that plasticity, that ability to respond physiologically to changes in CO2 is not free. An example being that if a polar explorer is walking through the Arctic or the Antarctic, they have mechanisms by which they can deal with some of the cold, the shiver, you change the way, the distribution of the blood in your body. But effectively, that comes at a massive energetic cost, and you consume thousands of calories a day just to keep yourself alive. So physiological plasticity is great, but it needs to be paid for. And we first kind of came across this concept way back now, 2000 and it was 2008, this paper was published. And when we started to look at ocean acidification, we just assumed that when you expose a calcifying organism to high levels of carbon dioxide, their rates of calcification would go down. That was the thinking at the time. So we ran an experiment on this organism here. This is a burrowing brittle starfish, Amphiura. Uses calcium carbonate to build its skeleton, and it's often been used to test responses of marine organisms to stress. So we exposed it to various levels of carbon dioxide, and what we actually saw was that the rate at which it could continue to calcify was maintained, and sometimes it was actually elevated in the high CO2 treatments, which came as a bit of a surprise, thinking, well, this is completely contrary to what we thought. But what was evident was that the dissolution or the difficult calcifying conditions were actually countered by this organism changing its biological deposition of calcium carbonate. It was actually upregulating that calcium carbonate process. So what we actually then saw, when you looked if you looked into the arms of this starfish, these are the arms. Here, this is a cross section of the arm. These are muscle bundles, muscle matter that you use to move the arms around. In the very, very high treatments, this muscle mass had completely disappeared. So what it was doing is it was energetically sacrificing the building of these muscles as it grew these new arms, because it didn't have enough energy to both create muscle mass and do the calcification. So there was a cost, and obviously for the implications for how that animal then functioned in using these arms to try and capture food, there would be a huge deficiency in how effective these arms were then capturing food. So really, even though if we just measure calcification, you thought actually this animal can cope, it can't, because it's sacrificing something else. And as we looked more into more and more organism responses, we saw more and more sacrifices in strange areas, such as the ability to respond to pathogens. So upregulating an immune system came at so they would deregulate their immune system in order to be able to then fuel other costs, which left them more exposed to infection. So when you're thinking about how organisms are going to respond to ocean acidification and environmental stress in general, really, it's important to think about the whole organism response. The physiological sort of structure of organisms is all meant to work together. So if we were to just run an experiment and measure an organism on one particular parameter, one particular response, we run the risk of actually missing what the effect is likely to be. And there are ways in which we can sort of holistically view organism responses to environmental stress. This is a dynamic energy budget, they were first developed particularly around looking at how bivalves responded in aquaculture, associated with a term called scope for growth. So the amount of energy that could be put into an organism's growth, how you could maximize that. But effectively, what that does is it looks at all the various groupings of physiological activity and understands how energy is distributed amongst those processes in the organism, to see where trade-offs are between general maintenance, between growth, reproduction, activity. And I think this offers a really useful framework in understanding how organisms respond to environmental stress, particularly in terms of acidification. We always look at mean responses as well in our experiments, don't we? But I think we're hearing about today is variability is key. For example, if we look at some of these data from real ocean acidification experiments, we see large sort of confidence intervals around the mean. And actually, I would argue that this is where the real information is. If we're thinking about how populations or species might acclimate or adapt to changing conditions, it's really in understanding this variability between individuals where we really want to be focusing our efforts. After all, we're all individuals. We all respond to environmental conditions in very different ways. Why would we assume that the animals that we run experiments on are all clones of each other and will give you exactly the same response? So start understanding, why is it an organism here that had very little response and this one had a huge response? And as we look more closely into this, we actually saw that there were even discrepancies between the sexes of individuals exposed to ocean acidification treatments, and that males and females responded very differently to the same treatments. And when you think about it, if it's an energy related response, then we can understand why that female part of the population that might be investing a huge amount of energy into the growth of eggs may well have less energy to be able to commit to responding to environmental stress than the males who are putting a much smaller energetic investment into the creation of sperm. So you can see that we need to understand why there's variability in response. This work off the coast of Chile sort of emphasizes what Helen said about the importance of understanding the local conditions of where you take your organisms from. Because what was being found was that in the early days of ocean acidification research, you take your organism from the field, and then you apply it to some generic level of ocean acidification, 7.9 or 8.1 or 7.8, without actually understanding that some of these populations are already living in 7.8 or 7.7. So what you think is a treatment is actually control for that organism. So the importance of linking the field data and the knowledge and the context of where these populations live to the experiments you run cannot be underestimated. And we've seen how ocean acidification can change biogeochemical shifts as well, in the same way that temperatures can drive biogeographic shifts, so can ocean acidification. And we can also see how populations of the same species, where they are distributed in their biogeographic range, actually affects their tolerance to some of these environmental stressors. So it becomes a very complex picture, but the take-home message is do your homework about where the animals are coming from in terms of an environmental context, what the animals are likely to be doing, what in terms of treating them as individuals, and understanding the sources of that variability that might impact on their response. So the points to remember, as I said, OA responses to OA in individuals intrinsically linked to energy. And that's something we need to remember, understand the energy, because it also means that some of the mitigating factors could be to provide organisms with more energy to deal with the stress. We've often seen in aquaculture species, particularly in oysters, there are some oyster strains that have been sort of genetically selected for by the aquaculture industry to maximize their ability to effectively use energy and therefore grow big. They actually show greater resilience to ocean acidification than other strains or other species that are and less energy efficient. And individuals and populations are not all the same. This variability results from experiencing different environmental conditions where they've grown up. And the intraspecific variability is also influenced by gender and life history stage. It's not something I've talked about today, but life history, organisms that go from, larvae through to adult phases. Not only can they be intrinsically different in terms of their vulnerability, but also they will live in different places. Experience times in different parts of the marine system, which might make them more or less exposed to ocean acidification. And experiments need to be mindful of all this, but I'm conscious of the time. So I want to move on to actually how do we go about monitoring this? Because what we've just seen means that there is so much to look at and understand. How can we possibly find indicators of biological change? We produced a paper, and it's something we've been kind of struggling with for a while. And a group, the GOAN Biological Working Group, has been kind of meeting up and sitting down in offices and arguing about this for many years, trying to find out ways to move forward. And we finally came up with a proposition for the community, which we published in this paper released last year. Effectively, the thing we're aiming for is to have some kind of globally coordinated effort to measure changes in seawater chemistry caused by OA, which we've pretty much achieved when it comes to the chemical changes through GOAN. There's a lot of resources in terms of how you go about measuring carbonate chemistry, how you bring those measurements together. But effectively, measuring carbonate chemistry, and Patrick will hate me for saying this, is relatively easy. You only have to measure 4 things, well, 2 or 4 things really, and these are the same things wherever you go. But when you measure biological change or biological impact, the choice is almost limitless as to what we can measure. So how do we collect biological information that can describe ocean acidification response, but actually can then be compared from place to place and from time to time, and it's relevant wherever we go? That presents challenges. So many different biological impacts of OA. What do you choose? And of course, not all biological parameters are important everywhere. There's no point me measuring coral in Plymouth because I don't have any. So coral cover's not really good for me. Not yet. Oh, yeah, we can, yeah, we'll have all of yours. Right. So, and of course, there's a massive range in local capability and capacity. So, if I was to come up with some fancy methodology that required £500,000 worth of instrument, that's not going to be very applicable to every marine station around the world. So it doesn't exactly bring everyone in. And we have to be mindful that monitoring the marine environment often isn't because we're going out looking at ocean acidification. Monitoring is often driven by other local interests and priorities. And how do we leverage off that and allow that to still happen, but then get added value? So what we've seen in terms of all the research we've done on ocean acidification in the past is that actually we do understand how organisms respond, but there are a number of key traits of organism behavior and physiology and ecological traits that kind of tie a lot of these responses together. And I'm conscious of time, so I'm going to go through fairly quickly. So as a group, we identified 5 areas where biological responses could be in some way linked to changes in ocean acidification. But remember, again, this is that because ocean acidification might not be the only environmental change that's driving these, but essentially, we have seen evidence that calcification, primary production, secondary production or growth, biodiversity and community structure, genetic adaptation can all be driven by ocean acidification, as well as other things as well. But there is a strong causal link demonstrated in laboratories that changes in those traits can be driven by ocean acidification. But absolute changes in impact data will be, how do we measure those? There's also countless ways to measure those traits. So it's important as well that we embrace that. We can't tell everyone to go out and measure calcification rates in a certain way. How do we allow people to measure in their own way, using their own skills and expertise, but still be part of the larger effort? And actually, bringing together diverse sets of data isn't the new problem. Meta-analysis on things like laboratory data have been doing it for a long time through a method called, looking at effect sizes. So basically collapsing the data down into sort of percentage changes. So it doesn't really matter the absolute amount of change, but it's the percentage of whatever you measured. And we thought we could actually do this in terms of ocean acidification data. If we think about time series, if we think about using time and change over time. So this is where the importance comes of having ocean acidification monitoring data alongside biological change data. If we want to progress along the proposal that we're doing. So what we propose is that if you can calculate your annual rate of ocean acidification change, the pH change, alkalinity, change in alkalinity, aragonite state, whatever, you can get a rate. You can get a rate of change. If you've then got some kind of biological trait that you've been observing, which can be related to one of the 5 traits we said before, and you've also been measuring that over time, you can then get a rate of change for that biological change. Thank you. And then, with those data, rate of ocean acidification change against rate of biological change, it gives us one of these dots on this graph. So we now have a mechanism by which we could say, if all of you had some measure of calcium of calcification, whatever it was, but you had a ocean acidification time series alongside it, we could bring all those data together on this graph. They would give us some kind of indication of what the general responses of calcification are across all these different contexts and different environments, and start to explore the nature of that relationship between calcification. And in fact, it will actually also start to give us insight into just how important ocean acidification is as the primary driver in some places, but maybe not in others, in some direct measures of calcification, but maybe not in others. So it's a way of bringing lots and lots of data together. So this is something we're looking to try and now undertake proof of concept. There's a Velux Foundation funded postdoc who's starting work in the University of Copenhagen, has just started. And she will be exploring through gathering together as many of these data sets as she can, just how robust a method like this is. So I think I'm going to end. My call is to everyone. If you feel you have a data set of biological change that you can relate to one of those attributes, those traits, and you have some supporting ocean acidification data and would like to see whether your data can be used as part of this proof of concept, I'd really, really would like to hear from you. Because currently it's very difficult for us to identify a strong indicator. We've talked about pteropods that are applicable in some places, a strong global indicator that we can attribute ocean acidification impacts to, and really understand ocean acidification's place in terms of a multi-stress environment. Okay, and I think that is, but finish with this one. So make sure, pay great attention to Patrick's teaching tomorrow, because without the carbonate chemistry alongside these data sets, it's very difficult to understand the true linkage between ocean acidification and the biological changes we're seeing all around the world at the moment. So thank you very much.
Maybe I could trouble you to control is your way of
starting my collaboration with my university of self classification as what in a lot of things. So we choose from developing and so we try to it's been like 10 years and we've small student. So although we are versus the lead, so we get this the gift from the source of the sand. Even though like 200 kilometers. And then we can see the changes of platform to platform leads and finally you can make the same time we also do TV videos statement videos from the began to convert the elements. But the interesting part is the recipe this is it's almost like really Hawaiian, but it's different from like it's closer to Okinawa bacteria. So there must be like a neuronal section of the bacteria whereby it's closer to the acidified condition. Then we have chosen that community. So we also in the genomics data and the classification data that like things we that I think not the productivity but the productivity is on the how much percent of technology living in the circles. Yeah, it's
Yeah, yeah, no, very interesting. And actually, that's so. I assume they're all members of the Icona group. Is that right? There's a yeah, Icona. Yeah. So we'll be reaching out to Icona definitely because whilst we've spoken about sort of time being the way in which you can do change, I mean, with the SEEPs gives us an opportunity to actually look at rates of change, but through space rather than time. But it's about collapsing down that data to the percentage sort of change in the environmental driver to the percentage change in the trait. So yeah, absolutely. We'll certainly be exploring how those gradients, be they spatial or temporal, could be useful for this.
I see. I think
I couldn't read out of the I think memory carbonate chemistries and grappling with my literally seven but points of things like I believe we were this more responsible than stock actually essential great all of the global compilations. And I think that one thing to keep in mind is that locally, and I think especially here, where we're comparatively in an area, we have quite a lot of data, but they're not really yet synthesized and brought together into large compilations for analysis. I think it's important to find that the local variability is really strongly driven by the biogeochemical variability that we have. And without understanding of dominance in our most renowned places like Sage or after topics, I think one has to be a little wary of going to global compilations that might, they give you data, but they might not reflect what is going on very well. And I think that really becomes important to understand when thinking about pharmacological impacts drivers down to if we're planning experiments here, what kind of treatments do we apply? Are they the right treatments? Do they reflect both seasonality or mean states of the carbon chemistry? So I think that's really one of us all together, bring our data together and bring our understanding
Yeah.
from different places what's going on, Thailand is different what's going on, here, Australia.
No, absolutely. My first piece of advice to any biologist doing an ocean acidification experiment is find yourself a biogeochemist who knows about carbonate chemistry. That's the most valuable thing you can do. Prepare to be inundated, Patrick, by biologists wanting to know about.
So we have one more question from online. Perhaps I can just repeat the question.
Yeah.
Biological trait sensitive to OA. Which category do you suspect would be the easiest to standardize across the different regions?
Do you want me to go first? Okay. I mean, I think calcification is probably the one that people would most go towards. I think it's the one that's probably got intrinsically the strongest causal link to changes in carbonate chemistry. I mean, many of the other traits, although they can be influenced by ocean acidification, they are going to be heavily dominated by warming and nutrient supply, salinity, all sorts of other things. And in those traits, it's very much about understanding the ocean acidification role in this larger picture of multi-stress environment. I would say calcification is likely to be the physiological process or trait that is going to give us kind of like where ocean acidification is going to be one of the major players. Did you want to? Nope. Okay. Hmm?
We have no other questions then. I think thank you so much once again, Prof. Widdicombe. Before